Good morning, everyone. For those of you that don't know me, my name is Rachel Gibson, and I'd really like to thank the organising committee for the opportunity to speak to you today on the gut microbiome. Now, I focused it down to the question, why does it matter in chemotherapy-induced gut toxicity? Now, I've specifically focused on chemotherapy. I'm not going to talk about radiotherapy or more recently the immunotherapies. In the interest of declaring conflicts, it's not going. In the interest of declaring conflicts, I have received consulting fees from Kaledo Biosciences. So the history of the gut microbiome in chemotherapy-induced gut toxicity extends right back to 1965. And recently, it's been identified in many papers that it has increasing importance as a key and fundamental player in the subsequent development of CIGT. Now, Frey and colleagues first identified this in 1965, where they noted that there was significant oral and GI shifts in myelosuppressed cancer patients. Now, remembering that this paper was published over 50 years ago, they had to rely on autopsy results from the first 50 patients that died from acute leukemia at the National Cancer Institute. And what they found was that nearly 90%, or 44 out of those 50 patients, had a major bacterial infection. They also noted that the bacterial infection had changed from the ones that were able to be treated with penicillin to a more staphylococcus infection. What they also noted was that the host defence deficits were proposed to be responsible for these changes in infection. In 2000, Nicole Blay-Levens and colleagues published a seminal paper which looked at changes in oral and GI toxicity for microbiome. And what they noted or proposed was that there was a phase known as the ulcerative bacteriological phase, which played key importance. Now, this paper was published 4 years before Steve Sonus's 5-phase oral mucositis paper. Now, what Nicole Blay-Levens and colleagues proposed was that it was the host defence mechanisms and the bacteria that really played a role, and the shift in potential between the commensal bacteria and the pathogenic bacteria, and really proposed that this was of key importance. Moving forward to 2013, Andrea Stringer and colleagues conducted a small clinical study which looked at 26 cancer patients that were hospitalised as a result of chemotherapy-induced diarrhoea. And what they found was that the majority of these patients that were hospitalised had significant decreases in lactobacillus, bifidobacterium, Bacteroides and Enterococcus, with subsequent increases in the pathogenic E. coli and Staphylococcus. Along with other changes, they proposed that this shift from the commensal bacteria to the pathogenic bacteria was responsible for the subsequent development of CIT. What we've noted is that there's significant changes in the microbiome, and this has really led to a suggestion that it plays a fundamental importance. So why do we have a microbiome, and why is it important? Well, we know that there's trillions of bacteria which exist right throughout the gastrointestinal tract, and different regions have different bacteria. If we look at the oral cavity, we know that this is a very complex ecosystem with moderate amounts of microorganisms. The predominant species which we see are Streptococcus and Actinomyces, as well as other obligate anaerobes. And we know that these changes can lead to disease, including periodontal disease. If we move forward to the stomach, we know that there's relatively low numbers of microorganisms, and this is thought to be due to the highly acidic environment. The most prevalent species that we know in the stomach are the Helicobacter pylori, and we know that these are responsible for the subsequent development of peptic ulcer disease in many patients. If we move forward to the small intestine, we still have relatively low numbers of microorganisms, and this is thought to be due to the presence of oxygen within the small intestine, as well as antimicrobials produced by the cells lining the small intestine, as well as the acidic environment. We know that the bacteria present are predominantly gram-positive bacteria. The most prevalent species that we see are the Lactobacillus and the Enterococcus faecalis. But it's really in the large intestine where we start to see the largest number of microorganisms, and this is particularly prominent in the descending colon. We know that there are different populations in the lumen and the mucosal regions, and the predominant species which we see are the anaerobic Bacteroides and Bifidobacterium, although we do see coliforms and Lactobacillus as well. So why do we have all of these microbiome and why are they important? Well, we know that they play an absolute key role in providing an exclusion or competition zone. So just by having commensal bacteria present means that there's unable for the pathogenic bacteria to colonise the gut. But they also play other key roles. So within our diet, we have a lot of fibre and indigestible starches. And it's these commensal bacteria which are responsible for breaking these down into what are known as short-chain fatty acids. These short-chain fatty acids are then responsible for a variety of different functions, including but not limited to stimulating the colonic epithelial cells to produce mucus, allowing the B cells that are found in the submucosa and the lamina propria of the gastrointestinal tract to produce immunoglobulin A, to be involved in tissue and wound repair, and this is thought to be particularly important in CIGT, promoting the production of Treg cells and immunological tolerance. There's also an hypothesis that these short-chain fatty acids are responsible for the inflammasome activation, the production of interleukin-18, as well as the maintenance of epithelial integrity. And finally, there's a thought that short-chain fatty acids are involved in the inhibition of inflammation and the key transcription factor NF-kappa B. But why is this all important and how does it all fit into CIGT? Well, in 2004, Steve Sonis produced the 5-phase mucositis model, and it's summarised in this diagram that you can see on the screen. Most of you will be very familiar with this model, where we have normal epithelium on the far side. We have the initiation phase, where chemotherapy comes in and produces reactive oxygen species, which then moves on to phase 2 and 3, where it triggers a whole range of messaging, signalling and amplification. In phase 4, the ulcerative phase, which is the most clinically relevant phase, we have a lot of bacteria being present before we finally have healing. And it's really in this ulcerative phase, phase 4, where the bacteria start to play an absolute key role. Because the epithelium is completely denuded, we have these bacteria which are now able to cross over, pass into the bloodstream, causing pain, cytokine amplification, as well as bacteria and sepsis for these potential patients. I'd like to take just a step back and talk about some of the really early preclinical studies which really started to suggest that the gut microbiome was fundamental in the development of chemotherapy-induced gut toxicity. And perhaps one of the best papers was one that was published in 1996 by Koyoshi Takasuma and colleagues. And this study showed for the first time that beta-glucuronidase, which is an enzyme produced on some bacterial cells, are really able to modulate intestinal toxicity following irinotecan. So irinotecan they administered to male Wistar rats at a dose of 60 milligrams per kilogram intravenously for 4 days. And you can see on the slide, here that these rats that received irinotecan lost significant weight and had substantial diarrhea and weight loss. However, what they were able to do was to give an antibiotic, and an antibiotic is able to inhibit the production of beta-glucuronidase, which is present on some of these bacteria found in the gut. And the rats that received this antibiotic in conjunction with this irinotecan had significantly improved diarrhea and significantly improved weight loss. This was really the first time that a study was able to show that bacteria were able to modulate the toxicity. This was then also shown in patients in a very small study by Köhler and colleagues in 2001. Now, this study only had 7 patients, but what they were able to show was that irinotecan-induced diarrhea was able to be significantly modulated by the administration of an antibiotic known as neomycin. And neomycin was able to significantly ameliorate this diarrhea in 6 out of the 7 patients, that's 85%. Now, recognising that the study is really small, they were also able to examine beta-glucuronidase, so that enzyme produced by bacteria, and they were able to find that this significantly reduced fecal beta-glucuronidase activity, as well as decreasing the fecal concentrations of that pharmacologically active metabolite SN38. But importantly with this study, they were also able to show that this didn't affect the efficacy of the chemotherapeutic drug. Moving forward to the mid-2000s, Andrea Stringer and colleagues conducted a variety of extensive research on changes between commensal and pathogenic bacteria following different chemotherapeutic agents. They used 5-fluorouracil, methotrexate and irinotecan. And the data that you can see on the slide here is from the irinotecan study. Now, this used female DA rats, and we know with female DA rats that following a single intraperitoneal dose of irinotecan, they have peak diarrhea occurring 96 hours, and they have peak weight loss also occurring at 96 hours. What Andrea did was she measured the fecal contents, and she looked at the presence of E. coli. Now, E. coli produces beta-glucuronidase, and we saw a significant increase at 96 hours, coinciding with that peak diarrhea and the peak weight loss. They also took samples throughout the gut and had a look at the expression of beta-glucuronidase using immunohistochemistry, and hopefully what you can see on this slide here, there's no brown staining, indicating very limited beta-glucuronidase, and quite intense staining seen at this 96 hours, coinciding with the increase in E. coli. They also had a look at the presence of lactobacillus, and you can see down the bottom here, there's very little. So this again was showing evidence that beta-glucuronidase was able to potentially modulate the toxicity that we're seeing. Other studies have also shown significant changes in gut bacteria populations following chemotherapy. In 2011, Zwillenheiner and colleagues looked at cancer patients with age, gender and lifestyle matched controls. There were 17 of these in an effort to look at the gut microbiome. Hopefully what you can see on this slide here is our cancer patients and our controls. So cancer patients, even before they underwent any treatment, had significantly less of the gut microbiome bacteria than their age, gender and lifestyle matched controls. Once these cancer patients underwent chemotherapy, there was again a significant decrease in their bacteria at 24 hours or 1 to 2 days after chemotherapy, and this only increased very slightly between 5 and 9 days. Importantly, one of the comments made by the authors in this paper was that potentially pre and probiotics may be an option for modulating this course. In 2017, Forsgaard and colleagues also looked at the changes that occurred in rats following a significant series of chemotherapeutic agents, and they looked at 5-fluorouracil, oxaliplatin and irinotecan. Now hopefully on this diagram what you can see is even compared to the controls that there were significant changes with all chemotherapeutic agents, irinotecan certainly had the most, with a significant change in the fusobacteria and the proteobacteria. Again, the authors of this paper suggested that probiotics was potentially a way in which they would be able to treat these rats before they underwent chemotherapy. So that poses a question: are we able to modulate the microbiome to enable effective supportive cancer care? So the idea of using probiotics in supportive care is certainly not new, and the mucositis study group from MASCC proposed in 2014 a suggestion that lactobacillus containing probiotics are able to be used for the prevention of GI toxicity in patients receiving pelvic radiotherapy. And this was based on the review of the literature and included 3 randomised control studies. However, it's an extremely narrow indication, and the panel were unable to make any further suggestions or recommendations other than this very narrow indication. Hannah Wardle and colleagues looked at a full meta-analysis in 2018 and looked at all new studies and came up with the idea that actually there is no benefit from using probiotics in these patients. So I want to pose the question, is it actually time to take a step back? So we know that the gut microbiome is involved. We've shown that in extensive studies right back to 1965. But I think it's actually quite critical that we now start to think more comprehensively and critically about the role that the gut microbiome has in chemotherapy-induced toxicity to enable us to guide intervention design. We need to really have a think about how we characterise these dynamic shifts in the microbiome relative to specific treatment milestones. And those milestones may be diarrhoea, it may be barrier dysfunction, or it may be infection. We also have the opportunity to identify unique microbial phenotypes, recognising that every person's microbiome is different, and this is different based on their gender, on their lifestyle choices, and on their diet, and can be associated with this desired response, which will allow us to use this clinical phenomena to drive our preclinical investigations and design. So I want to pose a question that can the gut microbiome be used as a risk predictor for CIGT? Now we know that there are unfortunately few very effective treatments, of any effective treatments for CIGT. But risk prediction for certain patient populations has previously been successful. Janet Collar and colleagues conducted a small study using genomic DNA where they looked at 34 cancer patients, and just under 30% of these patients had severe CIGT. What they were able to do was identify genetic variability in TLR2 and TNF-alpha, along with the cancer type, which was gastric or colorectal cancer. And they found that this was predictive of patients which were going on to develop severe CIGT. Importantly, this was both specific and sensitive, with ROC of 87.3%. So knowing that risk prediction is actually possible, I want to pose the question that can the gut microbiome also be used as a risk predictor? So we know, and it's very well established, that chemotherapy does cause many changes to the gut microbiome, and you just have to look at the literature to see that this is actually the case. We do know that perhaps the microbiome can regulate each individual's risk of CIGT, and Hannah Wardle and Wim Tissing proposed a new concept in 2017 of pre-treatment microbial profiling. And they proposed a comment that new risk stratification method and possibility of identifying patients early and those patients that are at high risk for developing CIGT. So what I'm going to present in just the last few minutes is some really early data that our lab has been conducting both here in Adelaide and also in the Netherlands. I'm only going to do a very quick snapshot, and I would really encourage you to get along to Hannah Wardle's presentation later this afternoon, where she's going to go into this in a lot more detail. I'd also encourage you to have a look at Kate Seckham's presentation later today and tomorrow. But what I really just want to give you a quick snapshot is that the aim of this study was to examine the relationship between pre-chemotherapy treatment microbial samples and the subsequent severity of CIGT. So this was a very small study that was conducted at the Royal Adelaide Hospital, where we recruited breast and colorectal cancer patients who were receiving a 5-fluorouracil-based treatment plan. Now, stool samples from these patients were collected both before they underwent chemotherapy and then again at day 5. And it's really important at this stage to recognise that none of these patients were chemotherapy naive. They'd all undergone previous cycles of chemotherapy. So the stool samples were collected and they were stored under appropriate conditions, and then they had their microbiome composition assessed by 16S parasequencing. And this was conducted at the Australian Genome Research Foundation. We were then able to access clinical case notes to assess each patient's diarrhoea, and this was assessed according to the National Cancer Institute Common Terminology Criteria for Adverse Events, version 5. Just so that we're really clear on the way that we assessed this, with the NCI-CT-CAE version 5, there are 5 stages, and the last stage obviously is death, which I haven't included here. None of our patients died from toxicity. So grade 0 is no change. Grade 1 is an increase of less than 4 stools per day over baseline. And so we grouped these together and classified these patients as non-toxic. Grade 3 and 4, the life-threatening and greater than 7 stools per 24 hours over baseline, we classified and grouped together as toxic. We excluded any patient that was classified as grade 2, and these were removed from all further analysis. So just to be really clear, our non-toxic patients were grade 0 and grade 1, and our toxic patients were grade 3 and grade 4. We showed no demographic differences between non-toxic and toxic patients. As I said, this was a very small study. We only had 12 patients in total. Of those 12 patients, 8 were classified as non-toxic, so had a grade of 0 or 1, and 4 were classified as toxic, so those grade 3 and 4. There are no differences in age, sex, cancer type, treatment protocol, and you can see down the bottom the number of different chemotherapy cycles that each patient had had previously. And just very quickly, what we showed was that baseline microbiome composition does drive treatment response, with Blautia being absolutely critical in determining the subsequent CIGT in patients receiving this 5-fluorouracil-based therapy. Hopefully what you can see on the graph in front of you is that our non-toxic patients, or our patients that didn't have any diarrhoea, have large amounts of orange, and this represents the Blautia genus. In contrast, if we have a look at our toxic patients, we can see in diarrhoea that doesn't exist. There's not as much orange and a huge amount of variation in the different genus species. When we had a look at absolute relative abundance, we found that there was a significant decrease between our non-toxic and our toxic patients in absolute relative abundance of Blautia, with a p-value of 0.081. So my take-home message from this presentation, what I hope I've shown you today, is that the gut microbiome is critical in shaping each individual's response to cancer therapy, with specific emphasis on chemotherapy-induced gut toxicity. And I really think it does have the potential to be further explored as a risk predictor. We need to conduct further in-depth longitudinal analyses in order for us to further understand the exact relationships with treatment milestones and outcomes. I think I've highlighted that baseline microbial profiling is likely to play a key role in risk prediction for patients at risk for severe CIGT, and our interventions do need to be guided by clinical phenomena. If you do want to hear more about this data, please pop along to Hannah's presentation and also Kate's presentation later today. And I just want to acknowledge the funding support that our labs have received over many years to conduct this research, most importantly from the Ray and Shell Norman Cancer Research Trust, Cure Cancer Australia, the Lions Medical Research Foundation, and the Flora Medical Research Foundation PhD projects. Thank you very much.